organs of the immune system, primary lymphoid organs. Now, our immune system is a very complex and highly defined system comprising concepts like innate immunity, adaptive immunity, concepts that basically look at humoral immunity or cell mediated immunity. And all these have been enabled because of the various organs in the body that take part or that are part of the immune system. So, this session is going to focus on primary lymphoid organs. Let us look at the outcomes of this session. So, the organs of the immune system are present at various places all over the body, and they are named as lymphoid organs because they have a role to play in production of lymphocytes. And lymphocytes are one of the cell types of the immune system that are actively involved in the immune response. The primary lymphoid organs comprise bone marrow and the thymus. The bone marrow is the site of hematopoiesis and B cell maturation. So, bone marrow is involved in what we term as it being responsible for humoral immunity because B cells are the ones that are producing antibodies, and antibodies are circulating all over. So, that is why that comprises the humoral immunity. Thymus is the site of T cell maturation, and it gives rise to a range of T cells, and these T cells comprise the cell mediated immunity. So you can see how bone marrow and thymus both are responsible for humoral as well as the cell mediated immunity. Now, just going or browsing through and or getting an overview of the parts of the immune system, when you look at the body, a human being, okay, you will find that all through the body there are various organs that are responsible for generating an immune response or being responsible for contributing to an immune response. So one can see that the bone marrow, the skin, you have the bowel itself, which has several lymphoid tissues that help in an immune response. The spleen just over the stomach. You have the thymus just over the heart. You have lots of lymph nodes forming the lymphatic system. So, lymphatic system is significant in the response or as part of the immune system. And of course, one has the mucous membranes in the nose and throat, the tonsils. These are all parts of the immune system which are helpful in an immune response or which are basically responsible in one way or the other to do that sole objective of ensuring that the antigen that comes into the body, antigen being the foreign molecule or the foreign organism that comes into the body, is eliminated before it can cause due harm to the body. Now, morphologically and functional evolution of the organs of the immune system begin early at the early embryonic stage itself. So, when it is in the yolk stage itself, one observed that you have the organs of the immune system, or rather the cells of the immune system being produced, and over a period of time, the organs are formed. Until early adulthood, development reaches maximum, and thereof it remains constant for quite some time, and that period is actually indeterminate. But once after a certain age, one finds that these organs, decline in their function, and so one observes, one always associates old age with compromised immunity or them being more prone to infections when you compare them to youth, okay, when you compare them to young people. After birth, the immune system is exposed. So, there is a certain level of development that happens until birth, but after birth, when the child or the child is exposed to different environments, and when exposed to different environments, it's exposed to different antigens, then it further contributes to the growth of the development of the immune system. Many organs, are actually just acting as barriers, which is important because They do not allow the antigens to enter the body in the first place. But then if the antigens do enter the body, then there has to be a system to take care of these antigens that have come into the body. And so there are many organs which actually give rise to the immune cells and the proteins. So therefore, of all the parts of the immune system, some act as barriers and some actually act as cells, as organs that give rise to cells and proteins that take part in the immune response. So the organs of the immune system is actually subdivided into two major groups. One is what is called as the primary lymphoid organs. They are also called as generative lymphoid organs because these organs are responsible for forming or initiating the process of forming the cells of the immune system. You have, for example, in these generating lymphoid organisms, two main organs associated. One is the bone marrow and the other one is thymus. The focus of this session will primarily be on bone marrow and thymus. And of course, one has the secondary lymphoid organs. And these secondary lymphoid organs are also called as peripheral lymphoid organs. And these organs are, responsible for proliferation and maturation of immature cells that have been produced by the bone marrow and thymus. And also the response is also when the cells are interacting or encountering an antigen from outside. So the several different secondary lymphoid organs include the spleen, the lymph nodes, the Peyer's patches, which is found in the intestine or the gut, adenoids, tonsils, MALT. This MALT stands for mucus associated lymphoid tissues. There you have several subtypes. For example, you have gut associated lymphoid tissues and so on and so forth. Now, what is interesting is that both bone marrow and thymus generate and produce cells that would take part in innate and adaptive immune system. Now, what is the basic difference between innate and adaptive immune system? So, innate immune system comprises cells, proteins, etc. that would, try to encounter any antigen that is coming from outside and try to eliminate it. Okay, so that is the first, immunity that, or the first defense that the body has against any entering foreign body. But if the antigen which enters the body prevails in the body, then the adaptive immune system will start producing cells that are very highly specific for that antigen and try to remove that antigen from the body. So, in a layman's word, an innate immune system is relatively less specific than an adaptive immune system because the adaptive immune system is especially developed to fight out a particular organism. And in the process of doing so, adaptive immune system also creates an immunologic memory. So once you have an adaptive immune system, adaptive cells formed, then those cells can remember the antigen probably for a lifetime. And so it is, effectively said that if a person suffers, say, from chicken pox once, okay, and the chicken pox is there in the person's body, the causative agent for chicken pox is there in the person's body for a long time, then the person is able to develop an immune response against it, which can remain in the body for a long period of time. So it is said once you suffer from chicken pox, you don't suffer from it again. Okay, the same can be related to chikungunya. It is said that once a person goes through chikungunya, then the person may not get chikungunya again. So basically, when you say may not get chikungunya again, it is just referring to the fact that when the causative agent enters the body, our system is already equipped to fight it out before the organism is able to create havoc in the body. So therefore, you can say that the body has got immunized against a particular pathogen. So that is where the adaptive immune system comes into picture. And innate is something which is the first response to any antigen, and that is equally important because you must understand that it is the innate immune cells that are able to initiate the formation of the adaptive immune system. So you must say that they go hand in hand again. So bone marrow and thymus are responsible for production of cells that are both part of innate and adaptive immune system, while the secondary lymphoid organs are mainly associated with adaptive immune system, because all immature cells, immune cells, when they encounter an antigen, they begin proliferating, they begin forming immunologic memory, they begin forming cells that are specifically able to bind to a particular antigen and get rid of it. So therefore all the secondary lymphoid organs are mainly associated with the adaptive immune system. Now we will focus, as the session focuses on primary lymphoid organs, we look at the bone marrow first. Bone marrow is basically the site of hematopoiesis. And this is something that is very important for the body. Now, interestingly, hematopoiesis involves the hematopoietic stem cells. And we all know that stem cells are cells that are undifferentiated and cells that have a special characteristic of self-renewal, and they have the ability to differentiate into many different types of cells. And this bone marrow is also a site of B cell maturation, so very much responsible for humoral immunity. Hematopoiesis initially by the stem cells begins in the yolk sac itself. Now, this is in the first trimester of gestation. From the first trimester of gestation to 7 months, the hematopoiesis begins to happen in the fetal liver and spleen. And by the time it reaches the 9th month, you will find that the bone marrow is now the site of hematopoiesis. So you can see how hematopoiesis evolves with the embryonic stages. So with every embryonic stage, what happens is that the hematopoiesis shifts to different organs, and then ultimately what is observed, especially in the humans and the mice, is that you have the hematopoiesis happening in the bone marrow itself. So like mentioned, hematopoiesis is process of formation of all blood cells. So therefore, hematopoiesis is a process of giving rise to not just immune cells, but also non-immune cells. And almost all cells that are present in the blood are formed through hematopoiesis. That's the name why the hematology, it's basically hematology, anything to deal with blood, that is why it is called as hematopoiesis. These hematopoietic stem cells are pluripotent. So when you use the word pluripotent, it basically means that it has the potential to differentiate into various types of cells. And the differentiation into various types of cells, of course, depends on the microenvironment that is present around the pluripotent stem cells and the growth factors and cytokines that would initiate differentiation into a particular cell type. So this is very important. Now, when you look at the hematopoietic stem cells, so just let us look at how hematopoietic stem cells eventually form the different cells of the blood, of which many of them are involved, or cells of the immune system. So, like mentioned earlier, hematopoietic stem cells have a special characteristic of being self-renewable. Self-renewal. That means they can keep forming more of itself, okay, without getting differentiated. So you can have more and more numbers of hematopoietic stem cells being formed, and you can see that all all through your life you will find that these hematopoietic stem cells are able to produce the blood cells all through. So these stem cells are extremely important. Now, along with self-renewal, we've already seen that the hematopoietic stem cells are pluripotent and they have the potential to divide into several different cell types. But the first differentiation under specific growth factors could be either formation of myeloid progenitor cells and formation of what is called as lymphoid progenitor cells. So the first differentiation or the first division, not division but differentiation of the hematopoietic stem cells is into myeloid progenitor cells and lymphoid progenitor cells. Interestingly, what will happen is that formation of the progenitor cells is such that it loses the capacity for self-renewal. Where the hematopoietic stem cells have the self-renewal characteristic. The myeloid progenitor cells and the lymphoid progenitor cells do not have the capacity to self-renew. That means they cannot divide to form more of themselves. They will have to come from the hematopoietic stem cells only. However, they have the potential to differentiate into further different types of cells. So from myeloid progenitor cells, you will get a specific cell lineage. When you say specific cell lineage, it means that these cells that are forming from the myeloid progenitor cells, belong to one family, and the specific cells that form from lymphoid progenitor cells belong to another family. So you will find differences in the cells of myeloid progenitor cells, and you will have differences in terms of structure, in terms of surface molecules on the cells, etc. So they are differentiated, and they differentiate into two different lineages. Now, this again will not happen without specific growth factors and cytokines. In fact, in the bone marrow, what has been observed that these hematopoietic stem cells actually grow and mature on stromal cells of the bone marrow. And these stromal cells are responsible not just for production of several growth factors and cytokines, but what is observed is also from external environment, a lot of growth factors and cytokines enter the stromal cells, and they nourish the hematopoietic stem cells and actually enable differentiation into different progenitor cells. So a microenvironment that is created because of the stromal cells helps in inducing the hematopoietic stem cells to differentiate into the progenitor cells. Now let us look at what the lymphoid progenitor cells are able to differentiate into. So they do form what is called as the dendritic cells. Dendritic cells are very much part of the innate immune system. So you can understand that the dendritic cells are cells that can actually address various different types of antigens, and not just one specific antigen. They are not highly specific, but they can encounter or they can interact with various different types of antigens. We will definitely have a look at what the function of dendritic cells are in coming sessions. Um, you can also have the lymphoid progenitor cells differentiating into what is called as natural killer cells. Natural killer cells are also part of innate immune system, and they too are able to address several different types of antigens. So the specificity is much less than the B cells and the T cells, but they are able to phagocytose, or they are able to not phagocytose actually, they are able to kill a lot of antigens through a specific process which would be taken up later. The lymphoid progenitor cells can definitely differentiate into B cell progenitor and T cell progenitor. So you can see that the lymphoid progenitor cells is able to differentiate into 4 different types of cells, and each of this can happen with respect to different cytokines binding to the lymphoid progenitor cells, and the lymphoid progenitor cells should have receptors for those cytokines and growth factors, and based on which growth factor or which cytokine comes and binds to the lymphoid progenitor cells, it will give rise to dendritic cell, or to natural killer cell, or to B cell progenitor cell, or to the T cell progenitor. So again, you must understand that here at this point of time, there are different transcription factors, there are different growth factors, etc., that give signal to the lymphoid progenitor cells to differentiate into a particular cell type. The T cell progenitor in turn can differentiate into T helper cell and differentiate into T cytotoxic cell. One also writes this as TH cells and this as the TC cells. And this both, that is the TC cell and the T helper cell, are comprising the cell mediated immune system. So it is these cells that are going to basically fight out the antigen or the foreign molecule that has entered the body. This gives rise to B cell. B cell is responsible for forming antibodies. So you can already see over here antibodies that are bound to the surface of the B cell, and each of these antibodies can be of different types, and they can have a specificity to bind to a particular antigen. So one B cell can recognize several different types of antigens. Okay, so B cell comprises the humoral mediated immunity because the encountering of the antigen is through a protein, which is the antibody, but the protein antibody is being formed by who? By the B cell per se. So you can already see how lymphoid progenitor cells are responsible for a response which is humoral or a response which is cell mediated. Now going to the myeloid progenitor cells, which is also derived from the hematopoietic stem cells. The myeloid progenitor cells can again give rise to dendritic cells, just like the lymphoid progenitor cells did. It can give rise to granulocyte monocyte progenitor and eosinophil progenitor, basophil progenitor, megakaryocyte, erythroid progenitor. So depending on what binds to the or what is the receptor present on the myeloid progenitor and what is the growth factor that binds to the myeloid progenitor, you can have these many different types of cells formed from myeloid progenitor. So you can see that it is pluripotent, right? Or you can say that it is multipotent because but it is not a stem cell because it has lost the capacity to self renew. Now, the granulocyte monocyte progenitor itself can give rise to two different cell types, monocytes and neutrophils. And these monocytes can further differentiate or further get matured into what is called as macrophages. Macrophages also are part of the innate immune system. Neutrophils are also part of innate immune system. Eosinophil progenitor can differentiate into eosinophils. Basophil progenitor cells can give rise to basophils. Megakaryocytes can give rise to platelets. And erythroid progenitor can give rise to erythrocytes. So you can see all the cells of the blood are formed to the myeloid progenitor and the lymphoid progenitor cells, of which all of these are part of the immune system, whereas these help in specific functions related to blood. We all know erythrocyte is responsible for helping in respiration process as it carries oxygen and carbon dioxide. Platelets are responsible for clotting, etc. So all of these have their specific roles, but you see they are originating from myeloid progenitor cells, which in turn are originating from hematopoietic stem cells, and all of this happen where they happen in the bone marrow. Now, of course, you would need a specific growth factor to enable the erythroid progenitor to differentiate into erythrocyte, or say you need granulocyte monocyte colony stimulating factor, which will help the granulocyte monocyte progenitor to get converted into monocytes or to neutrophils and then to macrophages again. So every which of these would need a certain growth factor that will help it differentiate into specific cell types. So what has been observed with blood cells is that there is a certain turnover rate. When you say turnover rate, what does it mean? It means that old cells die or old cells get degraded and new cells are formed. So there is a constant cycle of new cells being formed and old cells being degraded or eliminated. So that is what is understood by a constant turnover, or you can also call it the homeostasis. Now, say for example, erythrocytes have a lifespan of 120 days. So every 120 days, the erythrocyte that was formed is going to get degraded, and in the meantime, new cell must have formed, which in another 120 days would get degraded. So that is a continuous turnover rate happening with respect to erythrocytes. Similarly, neutrophils will have a lifespan of a few days. So neutrophils are formed and degraded. That process goes on, which means that because there is a constant turnover rate, this has to be regulated. So the regulation is by growth factors, by of course expression of receptors on the surface. So for example, unless the erythroid progenitor does not have a receptor for erythropoietin cannot stimulate the differentiation of progenitor into erythrocytes. So how much of receptors are present on the surface of the cell, that too can determine the actual constant turnover. Now, we've seen that the old cells tend to get eliminated or degraded, so therefore, apoptosis is something that is involved as a process of regulating the homeostasis. So, these are important facets, and every stage you would have different growth factors or cytokines functioning, and many of these studies have been carried out in humans and also in mice through knockout mechanisms. So, knockout mice have been used where either specific genes have been knocked out, and therefore based on the gene that is knocked out, what is happening to the mice, okay, or to the process of hematopoiesis. Such studies have enabled find out which are the growth factors involved in a particular step, or which are the cytokines involved in a particular step. Okay, interestingly, what has been observed is, for example, macrophages, which are formed by hematopoiesis, they themselves are maybe responsible for phagocytosing erythrocytes or eosinophils to maintain the number. Okay, so you can see how a product of the same process is able to take care of, a constant turnover of or homeostasis. Now we look at the fact that bone marrow is the site where B cell is developed and maturing. Okay, it's not just where you have the immature B cells being formed, but immature B cells getting converted into mature B cells. And this has been mainly, so the site of origin and development of B cells is mainly in the humans and mice. The immature B cells, they interact with the stromal cells, and the stromal cells secrete various cytokines, by lieu of which you have B cell selection. Now, what do we mean by B cell selection? So, we've all understood that the B cell on its surface has antibodies present, and these antibodies should be able to bind to foreign particles or foreign antigens that have entered the body. Now, what if you have a B cell which has formed but has no antibodies on its surface? So basically that B cell is not going to help the immune system in any way. Or say, for example, the B cell has been formed in such a way that the antibody on its surface is able to bind to one of its or one of the body's own cell or own protein. Then what can happen is that the B cell, instead of binding to a foreign particle, is binding to a self particle. In such a scenario, it is that our immune system is actually degrading our organs or our tissues or our cells. Now, that is detrimental, and so such kind of a situation or such kind of a process also leads to what is called as autoimmune diseases. Okay, so therefore, it is imperative that in the bone marrow, the B cells that would circulate in the body have to be those that have antibodies that may bind to foreign antigens, but you should not have self-reactive B cells proliferating. And so these self-reactive B cells have to be eliminated by apoptosis, which takes place in the bone marrow. And this process is what is called as B cell selection. So by lieu of this, one example that I can give is, say for example, insulin dependent diabetes mellitus. So the body is able to produce antibodies that will go and bind to the B cells of the islets of Langerhans of the pancreas, and because you have an antigen antibody complex being formed, the entire immune system is going to try to destroy the beta cells of the pancreas. And so what is going to happen is that the beta cells of the pancreas will get destroyed by our own immune system, and because of that the pancreas is not able to synthesize insulin, and because the pancreas is not able to synthesize insulin, the person suffers from insulin dependent diabetes mellitus. So you can see how insulin dependent diabetes mellitus can actually be an autoimmune disease. And that has happened because the B cell that is self-reactive to the beta cells were not eliminated in the bone marrow. So you can see how B cell selection is a very important process that happens within the bone marrow. Okay. Now, in different species, B cell development takes place in different organs, not necessarily in the bone marrow. In birds, it takes place in the bursa glands. In fact, the denoting of the B cells as B cells came from the word bursa, because the first time these B cells were identified, they were identified from the bursa glands of the birds. Okay, but fortunately or coincidentally, even in humans and mice, the B cells matured or developed in the bone marrow. So the B still remains, because B can be for bursa, B can be for bone marrow as well. In rabbits, it takes place in gut-associated lymphoid tissue. In rodents, it is taking place in the Peyer's patch. So the maturation of the B cells is happening in different glands, not necessarily in the bone marrow itself. All the hematopoiesis may be taking place in the bone marrow itself. So the immature B cells may be formed in the bone marrow, but in these species, the maturation of the B cells takes place in different secondary lymphoid organs. Now we come to thymus, where you have the T cells maturing. So thymus is basically present. Please do note that T cells, that is the T lymphocytes, the naive or the immature T cells are actually formed in the bone marrow. But these T cells migrate to the thymus, okay, and within the thymus, the T cells mature into different T cells that are able to encounter different antigens. So what is the thymus? Okay, the thymus is located just above the heart, okay, and between the trachea and the heart per se, and it is a bilobed structure. So you have the right lobe and the left lobe, and these two are interconnected by connective tissue, which is called as trabeculae, and also the trabeculae are able to differentiate within the lobes specific regions into lobules. Now, interestingly, the entire thymus gland is capsulated, so there is a capsule around it, and you have every lobule separated from each other by what is called as septae. Now, when one takes a cross section of the thymus gland, you can observe that this is how it is present. So this is one lobule, and within the lobule, again, when the staining is done, one finds that a certain region is more highly stained than a certain region. This highly stained region is what is called as the cortex, and the less stained region is what is called as the medulla. Now, the cortex is very dense, and therefore a lot of stain is taken up. That is because the cortex comprises innumerable amount of numbers of immature T cells, which are called as lymphocytes. So when the T cells enter the thymus, they are basically lodged in the cortex region, and here, depending on the microenvironment, the T cells can mature into effector or active or mature T cells per se. Looking at that a little more in detail, this is the lobule section of the lobule, where this part you can see is the medulla, and this is the part that forms the cortex. You'll find that there are a large number of thymocytes. All these blue cells that you observe are the thymocytes. They are the immature T cells, okay? And you would also find that in the cortex itself, there are several dead cells of the thymocytes that are present. And these dead cells have to be removed because they are not going to mature into T cells. So therefore, or T cells that can be circulated in the body. So therefore, these dead cells have to be removed per se, and they are removed by macrophages that are present. So these are the macrophages. Also interesting to note is that these thymocytes have to be getting differentiated into active or mature T cells, and that is aided by nurse cell, cortical epithelial cell. You have what is called as interdigiting dendritic cell. All of these, or the medullary epithelial cells, or the Hassel's corpuscles, all of these are actually helping in T cell maturation by forming specific growth hormones. So these thymocytes are actually converted into several different types. When you say T cell repertoire, it means that you can have different T cells with different T cell receptors present on its surface, and these different T cell receptors are able to bind to specific antigen MHC complexes. Now, what is MHC? MHC is major histocompatibility complex, which you will understand in detail later, but it is always that these T cell receptors are able to bind to antigens only when they are bound to the MHC. So that is something that is important. So every T cell would have a different T cell receptor that can bind to a different antigen with the MHC. So you have a diversity of T cell receptors that are formed thanks to the different hormones that are produced by all these different cells. So these cells, again, what is interesting to note, just like in bone marrow where B cell selection was carried out, in thymus T cell selection is carried out. So if there are T cells that are formed that cannot recognize any antigen, or T cells that are formed that are able to recognize self antigens or self molecules, then they should not come into proliferation, or they should not come into circulation, and so within the thymus itself they have to be eliminated, and that elimination is done by apoptosis. So that is how the T cells are selected. Now, it is interesting to know that with age, the thymic function decreases, and that is mainly related to the fact that the size of the thymus decreases with age. So, where it is about 70 grams in an infant, it goes to being only 3 grams in the elderly. So, what happens is both the cortex and the medulla shrink, and in actuality, the thymus is now containing more of fat rather than cortex and medulla, and so therefore the functioning of the thymus is going to be much compromised with age. Now suppose you have a person who has a non-functional thymus, then obviously what has been observed is, and of course many of these studies have been carried out in vitro where mice have undergone thymectomy. So thymectomy is a process where surgically thymus is removed, and so in such mice, what has been observed is that there is no maturation of T cells. The other type of mice that have been used are also called as nude mice. So in nude mice, what is done is that the thymus is irradiated, and because the thymus is irradiated, it becomes non-functional. So there the thymus is not removed, but it is made non-functional. So such nude mice are also not able to have their T cells matured, and so what is understood is that the cell mediated immunity is compromised, and because the cell mediated immunity is compromised, the mice can undergo, or they are more prone to infection, as circulating T cells are going to decrease. So this is something that is also observed in AIDS patient, because the HIV is actually the HIV, that is the AIDS virus, is able to enter or stay within the T cells, and when the AIDS virus multiplies, the T cells get destroyed, and so the number of T cells in an AIDS patient is going to be decreasing over a period of time, and therefore you will find that AIDS patients suffer from lots of secondary infection actually, and most AIDS patients do not die because of AIDS per se, they die because of some other infection. So they may not be able to fight out tuberculosis causing agents, they may not be able to fight out other pathogens, and therefore they would die from a secondary infection rather than from the AIDS itself. Okay, so a functional thymus therefore is very important, and with age, because the thymus is decreasing in size, you will find therefore that the cell mediated immunity slowly becomes weaker with age. So let's make the conclusions. Bone marrow and thymus comprise the primary lymphoid organs. Hematopoietic stem cells in the bone marrow differentiate to give rise to different cell lineages, which include RBCs, WBCs, and platelets. The bone marrow is the site of development of B cells, with selection of B cells that circulate in the body giving rise to humoral immunity. Immature T cells mature in the thymus to give rise to diverse T cells that circulate in the body, giving rise to cell mediated immunity. Most study towards understanding the functions of bone marrow and thymus have been done in mice. Therefore, primary lymphoid organs give rise to the cells of the immune system that result in humoral as well as cell-mediated immune systems, not to forget innate and adaptive immunity as well. Thank you.